Hi everybody, this is Mr. Bishop and welcome to this weekend screencast. We'll probably break these into two. I'll see how they go and we will go from there. But there's about 20 slides to get through, so probably two. We'll pause when there's a good time to pause. So, where we picked up in from class, we introduced Gregor Mendel. He is the first to study how traits or characteristics are passed on from parent or to offspring. He laid the foundation for modern genetics. Genetics is the branch of biology that studies heredity. Mendel is considered the father of modern genetics. Mendel's conclusions from his experiments with his pea plants. Each trait was controlled by two heritable units. Let's adjust these slides so you can see. Slide this image over. All right, now we can see. Two heritable units. We know this to be two homologous chromosomes. Each two copies of genes or alleles on each of the chromosome. The plants could have two alleles for shortness, two alleles for tallness, or one allele for tallness and one allele for shortness, depending on their parents' genetics. So we have a parental generation true beating for trait. We have a homozygous dominant, homozygous recessive, results in a heterozygous offspring. We breed those heterozygous offspring from the F1 generation, and we can get tall or short again, depending on the outcome of the genetic crossover. So this all happens at the genome, the genetic level. All of an organism's genetic material is their genome. For humans, this means all of the information contained on 46 chromosomes codes for us. As we saw with our dog activity, the code determines our genetic, our phenotype, what are physical traits. Traits are inherited characteristics encoded in that DNA, that recipe or instruction manual for building humans or building dogs or building flowers or pea pods. The inherited characteristics encoded on DNA, they include eye color, leaf shape, tail length, flower color, etc. Genetics is the study of biological inheritance, patterns, and variations in organisms. This leads us all to the law of dominance. So we've talked about dominant traits in class. When organisms having pure contrasting traits are crossed, their offspring will show one of the traits. The dominant, the recessive trait. The dominant will be the one that shows. The recessive trait does not show up. So if we have someone with brown eyes and blue eyes and we cross those over, the brown eyes will be dominant over the blue eyes. The dominant trait is tallness, black hair. Examples of dominant traits that we know of are tallness, black hair, brown eyes. We express those with capital letters, AA, BB, or heterozygous combinations. And recessive traits, short, blonde hair, blue eyes, are lowercase letter pairs. Dominance. One of the alternate possible versions of a gene. Alleles can be dominant or recessive. Dominant trait, the capital B, blocks out the recessive trait, lowercase b. Brown hair is dominant over blonde hair. So we have a pair of chromosomes, and we have the gene loci on the chromosomes represented by these strips. So this PP is a homozygous dominant trait, dominant allele. And then we have two lowercase a's, so that is homozygous recessive alleles. And then we have a dominant pair and a recessive, or dominant allele and a recessive allele, and that is a heterozygous. This can happen on the chromosomes. It just depends upon the parental's contributed chromosome or genetics. So dominant and recessive alleles. Dominant trait will be expressed if present at all. The recessive will only be expressed when two copies are present. So to have recessive traits, you need to have two copies of that gene. Example here, we have purple and white flowers. Purple is dominant. So purple will show up even if the offspring of a purple and a white flower, since purple is dominant, all the offspring will be purple. So Mendel and the law of segregation. So organisms inherit two copies of each gene, one from each parent. So we've done this. This is our monohybrid cross. So we have one copy of each allele from each parent, and we cross those over, and organisms donate only one copy of each gene in their gametes. Two copies of each gene separate during gametogenesis, the formation of gamete. Gametes are reproductive cells. They are sperm in men and eggs in women. So this is independent assortment, the process of random segregation and assortment of chromosomes, which results in the production of genetically unique gametes. So the chromosomes you receive from your parents will be some combination of their chromosomes from their parents, your grandparents, but they will not be exact copies of your parents' chromosomes. This is because of random assortment. Then when the sperm fertilizes the egg, there's a combination of your parents' chromosomes which constructs the genetic code for you. So alleles, alternative forms of a gene that may occur at specific locus or loci if we're talking about multiple. Each parent gives one allele to its offspring. So this is the allele for purple flowers on the top chromosome. That's the locus for the flower color gene in these chromosomes. This is a homologous pair of chromosomes, and but the alleles are different. So a pair of chromosomes is they match each other. They are the chromosomes that match up when you receive one chromosome from your mom and one chromosome from your dad, and they pair up depending on different biochemical makeup. Allele combinations. Every human somatic cell has two copies of every gene. The homozygous, both copies are the same allele, so that can be homozygous dominant or recessive, as seen in the allelic pairs there, or heterozygous, two different alleles. Again, we have the example of the homologous pair of chromosomes, purple flowers, white flowers, the loci on that pair of chromosomes. Phenotypes and genotypes. We've discussed this in class as well. If you're not clear on this, phenotype is the physical appearance, how you physically appear, whether you are tall, short, black hair, blonde hair, brown hair, blue eyes, brown eyes. Your physical appearance is your phenotype. Physical and phenotype both start with a PH. This is a good way to remember that phenotype is your physical appearance. Genotype, the word gene is in genotype. The genotype is the combination or makeup of your genes. Your genotype determines your phenotype, but you can have genes of both traits. You can only express one of those. Mutations. Mutations, as we've talked about during our discussion of evolution, is any change or random error in the DNA sequence. So Mendel's experimental cross. Mendel used pollen to fertilize selected pea plants. This is the P generation crossed to produce the F1 generation. So here is the example of flowers. The male sex structure is the stamen. The female sex structure is the pistil and flowers. We have purebred white and purple plants were crossed to create the F1 generation. Monohybrid cross examines the inheritance of only one trait. Example, flower color. Homozygous dominant crossed with homozygous recessive, all dominant phenotype. So we've done the monohybrid cross several times in class. Then we have the Punnett square. In 1905, Reginald Punnett developed a shorthand way of finding the expected proportions of possible genotypes of the offspring from a cross. If you know the genotypes of the parents, you can predict the possible genotypes of the offspring. So Punnett squares are our system for predicting all possible genotypes and phenotypes. Here's our monohybrid cross again. It is a grid system for predicting all possible genotypes resulting from the cross. We've done this several times. If you would like to do these again for more practice, there's many resources available online. Then we started on the dihybrid cross on Friday. This is a cross involving two different traits from two guinea pigs, hair color and eye color. The father and mother are heterozygous in their genotype for both these traits. So when we cross two heterozygous individuals, we are going to get a result of what happens when two traits are considered. You cross two F1 guinea pigs who are both heterozygous. You consider what types of gametes these animals can produce. 4 possible gametes: the capital B, capital S, capital B, lowercase b, capital S, and lowercase b and lowercase s. These are 16 possible combinations, 16 possible resulting offspring genotypes. Phenotype results follow from the Punnett square. The following phenotypes are possible: 9 brown hair, dark eyes, 3 brown hair, light eyes, 3 white hair, dark eyes, and 1 white hair, light eyes. So 9 dominant, 3 dominant recessive, 3 recessive dominant, and 1 recessive recessive. These ratios are not exact in reality because using Punnett squares to predict is like flipping a coin. You can predict the probability, but that doesn't always occur that way. And finally, we'll close out the screencast with a discussion about incomplete dominance. So sometimes the dominant trait does not completely mask the recessive trait. This is an example when red flowers are crossed with white flowers and they produce pink flowers. So here we have our pink flower in the middle. This is our incomplete dominance. All right, that is our notes for genetics. I hope you have a great weekend and enjoy yourself.